Our next topic is antimicrobial proteins, which enhance innate defenses by attacking microorganisms directly or hindering microorganisms' ability to reproduce. Now, the most important antimicrobial proteins, especially to the second line of defense for the innate defense system, are the interferons and complement proteins. Interferons can be abbreviated IFN. They're a family of immune modulating proteins, and they're actually able to warn neighboring cells because cells infected with a virus can secrete interferons, and these interferons secreted from the cells that are infected with the virus then go to cells that are not infected. Those are the healthy neighboring cells, and they warn them. They warn them that, hey, I'm your neighboring cell. I've been infected with a virus. You should do everything you need to protect yourself. And so interferons, when they enter the neighboring cells, are able to stimulate the production of proteins that block viral reproduction and degrade viral RNA. And that is going to be able to inhibit the infection of neighboring cells by viruses to an extent. Interferon type A or alpha, or B, which is beta, work that particular way. So it's interferon alpha and beta that work to warn healthy neighboring cells when a cell nearby has been infected with a virus. But there's also other types of interferon. Interferon alpha and beta, by the way, are also able to activate NK cells. Another type of interferon is interferon gamma. Kind of looks like interferon Y, but it's also called the immune interferon because it's secreted by lymphocytes. This interferon secreted by lymphocytes has widespread immune mobilizing effects and is able to activate macrophages. So interferons activate NK cells and also macrophages. So they're able to essentially indirectly fight cancer. There's also the fact that we've researched interferons, and now we are able to generate artificial interferons, which are used now to treat disorders such as hepatitis C, genital warts, and multiple sclerosis. Here in figure 20.5, we can see the interferon mechanism in action. This interferon mechanism is against viruses. So you can see at step 1, when a virus enters a cell, this cell over here is a goner. It's been infected. This virus is going to use the cell's own machinery to replicate its DNA. It's not only going to replicate its DNA, it's going to generate more of the capsules that cover its DNA. And so essentially what a virus-infected cell has become is a virus factory. But when the virus enters the cell and replicates, even though this cell is a goner, it switches on interferon genes. The interferon genes cause the production of interferon, and when the cell secretes interferon, that chemical can travel to nearby cells and bind to interferon receptors. The interferon binding stimulates the cell to turn on genes for antiviral proteins in its nucleus, produce these antiviral proteins, and these antiviral proteins block viral reproduction. So host cell 1, which has been infected by a virus, makes the interferon and is killed by the virus. Again, host cell 1's a goner. Once it's infected, it's infected. You can't save a virally infected cell. Whereas host cell 2 is binding the interferon, the warning molecule. It's being told about the virus from cell 1. It's being given interferon by cell 1, and the interferon is inducing the synthesis of protective proteins, antiviral proteins, in cell 2, so it can protect itself. So that's interferon. We also want to talk about complement. The complement system consists of about 20 blood proteins, and they circulate in the blood in an inactive form. This includes proteins such as C1 through C9, factors B, D, and P, and other regulatory proteins. They provide a major mechanism for destroying foreign substances, which we're going to get into, and their activation enhances inflammation. So that's going to be one of the big things we see, that the use of complement enhances the processes of inflammation, those same processes that lead to redness, heat, swelling, and pain, but also help promote healing. We also see that the activation of complement can directly destroy bacteria because it both enhances the innate inflammation, but also adaptive defenses. Now, the complement pathway can be activated a couple different ways. So essentially what a complement pathway is a way to activate certain complement proteins. Now, there's 3 different ways, but they're all going to lead to the generation of the same chemical. So we'll see that, but let's look at each one. So there's the classical pathway. This is when antibodies first bind to invading organisms and then bind to complement components, and that activates them. So there's a double binding process that occurs in the classical complement pathway. And once the initial complement proteins are activated, the activation cascade is triggered, so we'll get the generation of that similar molecule. The lectin pathway is going to involve lectins, and they're essentially kind of like glycoproteins. They're sugars produced by innate systems, and they're going to be able to recognize foreign invaders. And when lectin is bound to a specific sugar on a foreign invader, it can also bind and activate complement. And that'll lead to the activation of the cascade, which leads to our common chemical. And then there's the alternative pathway. And this is where we get the complement cascade activated spontaneously when certain complement factors bind directly to a foreign invader. This doesn't really have any inhibitors on a microorganism surface, so it generally just allows the process to proceed if we can get a complement factor to bind directly to that foreign invader. Now, no matter which pathway we use, 1, 2, or 3, classical, lectin, or alternative, no matter which pathway, the activation of proteins for complement is going to go in an orderly sequence. They're all going to catalyze each other kind of in a cascade fashion, and they're all going to lead to the generation of protein C3. Once C3 is made, C3 will be broken in two or cleaved into C3A and C3B. So splitting C3 is going to initiate the terminal pathway, or the last step of complement activation. And this is the same. The terminal pathway of complement activation is the same for all 3 pathways that activate complement, because they all converge and generate C3, no matter which one you use. And so the second half of complement activation, no matter which pathway you use, is the same. And that's what we're calling the common terminal pathway, which begins at the splitting of C3. So let's talk about what happens when we split C3. Well, C3b binds to target cells, like a microbe, and that actually triggers the insertion of complement proteins. So these are proteins that are called the membrane attack complex. So these are essentially proteins that insert themselves in a sequential order in the cell membrane of a microbe and create a pore. So the MAC forms and stabilizes that hole or that pore in the membrane of the microbe. And because there's a pore, now we can see, or what can occur is the influx of water. And when water influxes quickly into a cell, lysis usually soon follows. And so the cleaving of C3 into C3a and C3b can lead to cell lysis because of C3b acting as a target cell binder. So C3b also causes some opsonization. So it can also act as a cell marker that can increase not only inflammation, but can increase the process of phagocytosis. C3a and then other complement proteins, they're going to be able to amplify inflammation by stimulating white blood cells like mast cells and basophils to release histamine. We know histamine is required for vasodilation, which is a hallmark of inflammation. We can also increase the mobilization of leukocytes or attract neutrophils and other inflammatory cells by using C3a as an inflammation amplifier. So if we put all this together, we can see that whether or not we use the classical, lectin, or alternative pathway, we will eventually create C3. C3 is a complement protein that, when broken into C3b and C3a, can lead to enhanced inflammation, opsonization, and cell lysis. The cell lysis is achieved via the use of C3b and membrane attack complex proteins. The membrane attack complex proteins embed themselves in the cell membrane of a target cell, which is our microbe, create a pore, and cause an influx of water, which causes cell lysis. Opsonization is the coating of pathogen surfaces in order to enhance phagocytosis. And the enhancement of inflammation is achieved by C3A and generally C5A. And they're going to work to not only increase blood vessel permeability and attract phagocytes and upregulate chemotaxis, but they're also able to stimulate the release of histamine. And now let's also talk about fever. So this is one of the last topics we're going to talk about for our second line of defense for the innate defense system. Fever is just an abnormally high body temperature, but it's systemic. So a lot of what we've been talking about was local. Now we're talking about something that's systemic, but is still related to the innate system. But it's a systemic response to invading microorganisms. So white blood cells and macrophages, when they're exposed to foreign substances, they secrete chemicals. Some of those chemicals are pyrogens. So I'm just going to stop right here so we can define pyrogens. Pyrogens are chemicals that cause fever. Pyrogens are able to cause fever by traveling in the blood to the hypothalamus. They act on the hypothalamus, which is the body's thermostat, to raise body temperature. And what are the benefits of this? Well, a moderate fever, not a severe fever, but a moderate fever actually tells the liver and the spleen to hold on to iron and zinc. And microorganisms or pathogens need iron and zinc. And if the liver and spleen are not releasing them, that's going to inhibit bacterial and other microorganism growth. Excuse me. We also know that fever or increased heat in the body can increase the metabolic rate, which increases the rate of repair or healing. So it's important to know that pyrogens are chemicals that cause fever, that fever is a systemic response. Most of the innate system is a local response, but fever is going to be a systemic response. And if it doesn't work, oftentimes we'll see the adaptive system be called in. It's good to have a moderate fever, not to have a severe fever. A severe fever can denature enzymes, which are proteins, and that can lead to decreased chemical reactions occurring in the body and even death in very serious circumstances. I also recommend looking at table 20.3, which just goes over the second line of defense, looking at those innate cellular and chemical defenses that we've been looking at, beginning with our phagocytes like neutrophils and macrophages, looking at our non-phagocytic natural killer cells, which use perforins and granzymes as their mechanism of action, looking at the inflammatory response and the 4 cardinal signs of acute inflammation: redness, pain, heat, and swelling. Looking at our antimicrobial proteins for the second line of defense, the interferons and complement. Be familiar with interferons and what they do, just be able to explain how the infected cell secretes interferons and that warns the neighboring cell. The neighboring cell will produce antiviral proteins to protect itself. And then with complement, know what it does. Know what happens after the generation of C3, and when that is cleaved, that not only are we going to promote cell lysis, but we're going to enhance inflammation, and also that we are going to enhance phagocytosis. And then know what pyrogens are. Pyrogens are the chemicals that cause fever by acting on the hypothalamus, which is the body's thermostat, and we would increase body temperature when the pyrogens act. And that is it for part A of the immune system. Make sure to watch the other parts, part B and part C.